In this lecture, we'll be discussing gene expression. This will be a culmination of the past 4 chapters that we've talked about. In chapter 24, we talked about genes. What are genes? Chapter 25, we dealt with how DNA is replicated, so how genes get copied when cells divide. Chapter 26 was dealing with transcription, so taking the DNA language and turning it into messenger RNA. We talked a little bit about how messenger RNA is also, there's some processing that goes into that, right, to get a mature messenger RNA. And then chapter 27, we dealt with translation, so turning messenger RNA into protein. And then we also mentioned in that chapter, there's post-translational modifications that can occur in proteins, things like phosphate groups get added, and so that's all part of it as well. So gene expression is kind of all of this stuff as a whole. So there's some of the key topics we'll talk about in this chapter. Elements that control transcription, those are DNA elements. Protein factors that can control transcription as well. Lac operon, we're going to be discussing. There's a lot of details in this chapter, and really I kind of want to do more big picture stuff here for this class, but the lac operon is one thing that we will kind of go into some detail and you'll be responsible for. It's a model control system, gene expression system in bacteria, and it's also used a lot in biotechnology, and so it's one of the more well-known ones. So that will be the one really specific thing that you'll be responsible for. Okay. And then there's also regulation of protein synthesis by RNAs, and we'll see some of that a little bit later in the lecture. Basically, gene expression in a nutshell, it's how much of a functional protein is present in a cell at any given time. So how do you control protein's activity in a cell? Well, one, it's how much primary RNA transcript or messenger RNA that you make, that you transcribe from a gene. 2, how you process that RNA into mature messenger RNA. How rapidly that messenger RNA can degrade, how long it sticks around. How much protein do you make from the messenger RNA? That factors into it. How efficiently to target the protein to its location. So if it's targeted for the mitochondria and it doesn't really get there very quickly or efficiently, that before it degrades, that would factor into the steady state amount of the protein in a cell. How to alter the intrinsic activity of the protein, so things like post-translational modifications. You can have some protein around, but if it's not in the active form, it's just sort of dormant. Okay. And then finally, how rapidly the protein degrades once it's made. And so when we look at these processes just in a picture form, that's shown here, right? So a gene you have in DNA, how much transcription you make, right? And how many primary RNA transcripts you make of that, that's part of it. Processing that into mature mRNA, how stable that messenger RNA is, how well it's translated into protein, how well that protein's modified, protein transport to get to where it needs, and then finally how long that sticks around, or is that protein degraded very quickly. Past focus of gene expression has been on understanding transcription initiation. Okay, but as you saw, there's other levels of control here for gene expression. Right? So there's increasing research going on in post-transcriptional and translational regulation as well. These mechanisms can be very elaborate and interdependent, and that especially in development. So when your organism is in a very early stages in quickly growing and cells are dividing, and their cells need to be turned into specific things. This gene expression is very, very important. Okay. The regulation is relying on protein DNA and protein-protein contacts, and we'll see a little bit of that. Right. Some vocabulary for gene regulation. A housekeeping gene. These are ones that we consider to be really always being expressed, okay, and in pretty much all cells. Some examples are shown here. GAPDH from glycolysis is an example of one of those, and beta-actin as well. So just as a little aside, if you're doing Western blotting in a lab and you're looking for a protein of interest, typically you'll also do a antibody for a housekeeping gene such as GAPDH or beta-actin, and that just kind of gives you a control to make sure you're loading the same amount of material on each lane. Okay. The other types of genes are regulated. Okay, so if they're not constantly expressed, they're regulated. Levels of the gene can rise and fall based on the needs of the organism. Okay. We call these genes either inducible, that be able to turn them on, or repressible, being able to turn them off. So RNA polymerase, as we saw in transcription, it binds to promoter sequences that are near the start point of that transcription initiation. This RNA polymerase promoter interaction is what's influencing the rate of transcription. Some regulatory proteins, known as transcription factors, if you remember those, the TFs, those work to either enhance or inhibit that interaction between the polymerase and the DNA promoter site. So we're going to break this down into a few parts here. So part one, bacterial gene regulation. E. coli promoters have sequences close to a consensus sequence. So most bacterial promoters have the conserved -10 and -35 regions. And that those, again, if you think back to that lecture and that chapter dealing with transcription, those are interacting with the sigma factor of the RNA polymerase. Right, substitutions in those regions usually reduce the affinity of RNA polymerase to the promoter. Some promoters also include what's known as upstream elements, and those interact with the alpha subunit of the RNA polymerase. Those are a little bit further away. So we've seen this picture before. The -10 region is known as the TATA box. -35 is the other consensus region, and then you can have upstream of that what are known as up elements. Okay. So hopefully that looks somewhat familiar. Okay. So the use of these sigma factors, these recognize different classes of promoters. So depending on what sigma factor, there are different sigma factors. They're going to recognize a certain class of promoters, and that allows coordinated expression of different sets of genes. Right? So not every gene has its own sigma factor, right? You have groups of genes that are doing a similar thing, are needed for a similar set of conditions that are under the control of a single sigma factor for that set. Now, binding of other proteins, those transcription factors to promoters, is the second part of this. Those recognize promoters of specific genes. They bind small signaling molecules, or they sometimes bind small signaling molecules. And they undergo post-translational modifications as well. The protein's affinity towards DNA is going to be altered by ligand binding or these post-translational modifications. And this allows expression of specific genes and responses to signals in the environment. So if we look at the regulation of the sigma subunits of RNA polymerase, these what's known as specificity factors. Those alter RNA polymerase's affinity for certain promoters. So the sigma subunit of E. coli RNA polymerase, that's an example of this specificity factor, right? Most of the promoters are recognized by that sigma 70, so that's kind of the typical one, or the most common one. It can be replaced by one of 6 additional specificity factors. So in heat shock conditions, when the temperature goes up higher, sigma 70 is going to be replaced by sigma 32, and that's again, sigma 32 is going to be used in different promoters, and those promoters are in front of genes that help out in higher temperatures. So, when the bacteria are subject to this heat stress, and that's typically over 40 degrees Celsius, sigma 32 comes in, and you get a starting expression of different proteins that will help in these conditions, and one of those are chaperones, or HSP, heat shock proteins. So number 2, we're talking about small molecule effectors that can regulate these activators or repressors. So what is a repressor? A repressor reduces RNA polymerase promoter interactions, or you can think of it in other terms, blocking the polymerase. Right? It binds to what is known as an operator sequence. So repressors bind to operator sequences in DNA. Operator sequences are usually near promoter in bacteria, but in eukaryotes, they can be very far away from the actual promoter sequence, and we'll see examples of that. Right? Effectors bind to repressors and induce a conformational change. So a repressor is a protein that binds to an operator site on DNA. Effectors can change the conformation of repressors. And that change can either increase or decrease that repressor's affinity for the operator. So thus it can either increase or decrease transcription. So activators improve contacts between the RNA polymerase and the promoter. Binding sites in DNA for activators are called enhancers. So an activator is a protein. The binding site in the DNA for that activator is called an enhancer region. In bacteria, again, enhancers are really close to the promoter. And in eukaryotes, they can be, again, very far away. And this brings up the difference between what we call negative and positive regulation. Negative regulation involves repressors. Right? Repressor, again, it's binding to the operator and it's stopping transcription. Positive regulation involves activators. So here we have an example of negative regulation. And you can have two ways here, right? You can have negative regulation where your repressor is in the normal state, bound to your promoter, and transcription's turned off. And then you have a molecular signal, right? Some sort of small molecule signal from the environment comes in, binds to the repressor, and that causes it to leave the operator, which turns on transcription. You can also have negative regulation where the repressor is normally not bound to the operator and thus transcription's on, but once it binds a molecular signal, then it binds the operator and turns off the transcription. Okay. And just how you can have two different, switching mechanisms there for negative regulation. You can also have that for positive regulation. In one, the activator is always, I shouldn't say always, it starts out being bound to the activator site or enhancer site, and then transcription's on. But once it binds a molecular signal, that removes the activator and thus turning it off. Or you can have a case where the activator needs to bind a molecular signal to get to that enhancer site and turn on transcription. So that just gets a little bit confusing here. Both of these are positive regulation, and on the previous slide, both of those were negative regulation, even though the mechanism turning it off or on based on your small molecule binding is different for each case. Okay, just a transcriptional regulation summary here. Transcription factors, those are proteins that bind DNA, regulate transcription. Those are activators or repressors. Okay. Effectors are those small molecules that bind to the transcription factors. They either increase the interaction or decrease it, okay, of the transcription factor with the DNA. So you can have both cases in activators and repressors. Operator, that's the region of DNA that the transcription binds to repress transcription. And an activator or enhancer region, that's the region of DNA that the transcription factor binds to induce transcription. So those terms will be important for the exam. It turns out that in eukaryotic cells, they follow a similar machinery, but it's just much more complicated. So in eukaryotic cells, and you're not going to be responsible for knowing really a lot of specifics here for eukaryotic cells, but if you go on to medicine or study biology, biochemistry in the future, these are probably ones that you're going to focus more on, I would assume. So it's good to have just a little bit of background on them. But the activator in operator regions in eukaryotic genes and probably don't really have operators. It's really more the activator enhancer regions for eukaryotic gene expression. But they can be located very, very far away from the promoter regions and the genes that they control. And one thing that how this works is that the DNA can bend, and you can, something that's in primary sequence in DNA very far away from a promoter can be bent and made into very close proximity to it. And that's just really what this is showing. And that's done through architectural regulator proteins that help bend it, and co-activators that help the enhancer or activator regions reach the promoter region. So many bacterial genes are transcribed and regulated together in an operon. Okay, so an operon is a cluster of genes sharing a promoter and regulatory sequences. So you can think of it as an operator that has many genes associated with it, or maybe not many, but several genes associated with it. And the example we're going to see and use as our specific example is the lac operon. Okay. So our promoter here and an operator sequence here, and then we have 3 genes that are just transcribed from that promoter. And if we look at this in a little bit closer detail, the operator sequence is shown here in yellow. Promoter is here in kind of a teal color, and we have 3 genes: LAC-Z, LAC-Y, and LAC-A. Right? What they encode for: beta-galactosidase, permease, and transacylase. Right? We have also this LAC-I, which is a gene that encodes for a protein that is our inhibitor, right? So that binds to the operator and actually represses the transcription of these genes. So this was, I believe, one of the first gene control mechanisms that was discovered, and that was in the 1960s. And the people that discovered this won the Nobel Prize. Okay? And again, there's three genes: LAC-Z, LAC-Y, and LAC-A. The beta-galactosidase from LAC-Z that cleaves lactose. So this is, we call this the LAC operon. This is all dealing with the metabolism of lactose, the sugar lactose. So beta-galactosidase cleaves lactose, which is a disaccharide, to yield glucose and galactose. Okay. And lactose permease, LAC-Y gene, transports lactose into the cell. Okay. Those are the two that important to know. All of these are regulated together as what we call an operon, and it relies on negative regulation via a repressor. So if you just curious here, that lactose permease is here, and then beta-galactosidase cleaves the lactose into galactose and glucose. I should mention too, I should have mentioned on that previous slide, it also can isomerase that lactose into a molecule called allolactose, and that's what becomes the effector for the repressor of that LAC operon. Okay. So how does this mechanism work? Well, when glucose is abundant and there's not very much lactose around, cells make only very low levels of these enzymes for lactose metabolism, right? If you have a lot of glucose around, that's easier to metabolize, and you don't have lactose, there's really no reason to transcribe these genes, right? You just waste energy. Okay, so in that case, transcription is repressed. If glucose is not around, low concentrations, but lactose is present, then the cells can use their energy to produce these enzymes to break down lactose and then get energy from lactose. So that is the case where transcription is no longer repressed. So how does this inhibition work? Well, that's that LAC-I gene, and that's in the very front of this region before the operator sequences. All right. In LAC-I, you can think of I as standing for inhibitor. That it encodes the LAC repressor. Okay. And it has its own promoter as well. So it doesn't have the promoter shown here that would promote the other genes. It has a separate promoter. Right. The repressor that it makes, that gene, that protein product that gene makes, can bind to 3 operator sites in this gene. Okay. The LAC repressor binds primarily to operator 1, right? And that's the one that's adjacent to the promoter. And binding of that repressor helps prevent the RNA polymerase from binding the promoter, so you block transcription. You can also bind, once you're on that first operator, the repressor can also bind one of the other 2 and make a loop there. Just kind of think of that as tying a knot there that further hinders the polymerase binding to the promoter site. Really, the important thing that I think you should know in this class is really just that the LAC repressor binds the operator sequence, thus blocking transcription. Less important that you know that there's actually 3 operator sites and that you can get these loop formations there. Okay. Another sort of cartoony picture, right? You can promote your RNA polymerase on that promoter site, make the LAC-I gene, express it, I should say, into messenger RNA that encodes for the LAC repressor, then that combine these other operator sites that blocks transcription. And it reduces transcription 1,000-fold. Right? This repressor, this isn't super important, but it's not a small molecule. It's actually pretty big. It's a tetramer, and it's actually a dimer of dimers. Okay? So if you're interested in that, this is sort of how it looks there. But looking at it in just a cartoony picture is probably fine for our class. The LAC operon here is what we call being induced. So it's an example of, in normal conditions, that repressor is bound to the operator and transcription is hindered. Once a small molecule effector binds to it, then that operator gets removed, the repressor gets removed from the operator and transcription can occur. And so the molecule that binds here is called allolactose. It binds to the repressor. Okay, so we call this an inducer. Binds the repressor, causes it to dissociate from the operator. Okay. So beta-galactosidase, that not only hydrolyzes lactose, but it also can isomerize lactose into allolactose. So that's where the allolactose comes from. It's an isomer of lactose. Okay. So the concentration of allolactose goes up when the concentration of lactose goes up. Okay. In the lab, the LAC operon is very commonly used in bacterial plasmids, expression vectors, where you want to insert a gene for a protein that you want bacteria cells to grow for you, so that then you can purify it. So those are most commonly used, the LAC operon. Okay? And instead of having the cells always express your protein, expressing the protein is energy intensive. So you don't want to have them constantly making this protein. You want to first grow the bacteria cells up in culture so that there's lots and lots of cells. Then you want to say, okay, now start expressing the protein. Because if they were constantly expressing some of these proteins, the cells just wouldn't grow as much, and you wouldn't make as much. Right? So how we induce these cells in culture, we don't usually use allolactose. Why is that? Well, it's because, right, lactose will be metabolized by the cell and it breaks down, and then the expression of the proteins would stop. Right? So instead, we use an artificial inducer, IPTG. So if you ever work in a biochemistry lab expressing proteins, there's probably like a 95% chance you're going to be using IPTG. Okay? And it binds that LAC repressor and removes it from the operon. And this is just an example of that from the lab. This is a vector I've used, which has the LAC operon or LAC operator here by the T7 promoter. And so this is the multiple cloning site. You put your gene of interest in there, and it's controlled by that LAC operon. Okay. And forget what all of these lanes are, but we'll see a better Western blot here in a second. I'm assuming this was kind of before I induced, and this was after I induced. Okay? And you'll notice that there's still a little bit of protein coming out before you induce with IPTG. It's called leaky expression, but obviously much, much more after you add IPTG. Right? And so this is another figure from a paper. So lane 1 is cells before you induce with IPTG. Lane 2 is after you induce with IPTG. So no protein expressed after IPTG, you start making that protein, and that's controlled by the lac operator. So it turns out that the lac operon is governed by more than just repressor binding. Ability of glucose, or availability of glucose, governs expression of these lactose digesting genes. And that's what's known as catabolite repression. So when glucose is present, lactose genes are going to be turned off, regardless of whether lactose is present or not. And this is mediated by cyclic AMP and cyclic AMP receptor protein. So that's CRP or CAP for catabolite activator protein. This is probably a little bit over the scope of the class, but cyclic AMP receptor protein, that's a positive regulator of the lac operon. CRP binds to the lac operon in the absence of glucose. So when there's no glucose around, this is binding there and sort of turning it on. So that's the opposite of the lac repressor. A region in this protein interacts favorably with the RNA polymerase, and that stimulates transcription of those genes. Cyclic AMP binds near the promoter, and that stimulates transcription about 50-fold. Okay? And how that does it, it's bending the DNA. And so the open complex doesn't really form very well without this CRP, cyclic AMP activator. This only works when the lac repressor has dissociated. Okay, so that's the other part. So the lac repressor kind of has precedence here. Cyclic AMP, if you're curious, that's a signaling molecule we talked about in the second quarter of the class. That's made when glucose concentration is low. So when glucose is low, this concentration goes up. It can start having this positive effect on this operator, and then once you have lactose present, that repressor can be removed and you can start transcription. So just how this looks in the picture, repression, you can have two states of repression, right, depending on if glucose is low or high. When glucose is low, right, remember we have cyclic AMP, so this activator is bound, CRP is bound, but the lac repressor is still bound, right, if you don't have lactose present, right, so you don't get any activation there, you don't get transcription. Once lactose is present, then the lac repressor gets removed and you can get activation. Okay. When glucose is high, meaning you have low cyclic AMP, you don't have activation here, right? And even if you add lactose to remove that repressor, if you don't have glucose, if you have a lot of glucose, you don't have cyclic AMP, or those cyclic AMP is low, you don't really get an activation there of the lac operon. Okay. So you really want to express these genes only when lactose is present and glucose is not, because the cell would prefer to use glucose as a energy source, because there's less steps involved in cleaving that disaccharide there. So when lactose is absent, you get no transcription because that lac repressor is still bound. Okay, so this is just the same picture, just a different way to look at it. When lactose is present, that transcription level is going to depend on glucose level. If the glucose is high and lactose is present, you're not going to get as much transcription. It's going to be a low level. When glucose is low and lactose is present, that's when you really get the high level of expression of those lac operon genes. Okay. So two requirements for that strongest induction of the lac operon: lactose must be present, that's when that's present, you get that allolactose. Repressor binds to the repressor, that repressor gets removed. And then glucose must also be low so that cyclic AMP can increase the CRP binding in the activator site. So you reduce the repression, yes, there we go. And then you also cause activation. Yeah, and this is, again, just a summary of the different conditions here. Lactose is low, your repressor is bound, you get inhibition. Lactose is high, that repressor dissociates, that can permit transcription. But then you also need to factor in glucose. So if glucose is high, CRP is not bound, transcription is dampened somewhat. When glucose is low, cyclic AMP high, CRP is bound, you get activation. So lactose is high, glucose is low, that's when you get the biggest activation. Okay, so that's our model system. Okay. Where do these proteins bind to the DNA? And for this, you're not going to need to know these specifics. This is, really just more for if you're interested. But it turns out they bind primarily in the major groove of DNA, right? So DNA double helix, you have what's known as the major groove and then the minor groove. So one side, and on a three-dimensional model, it's a little bit easier to see. But the major groove here is bigger than the minor groove. Okay. The exact specific hydrogen bonding interactions, again, not super important. It turns out that the major groove is a good size for an alpha helix, right? So that's a secondary structure in the protein that can interact there. There are a few protein motifs that turn up in these activators or repressors. And some of them you might have heard before. Something like zinc fingers, I think, are fairly common. Helix turn helix is another one. That's the one that's used by the LAC repressor. And then another type is called the leucine zipper. So just looking at these, knowing those three is probably good for the class. Maybe knowing the specifics of them all, not necessarily the biggest deal. Okay. So I'm going to kind of go quickly through these. So the helix turn helix motif, it's about 20 amino acids, one helix for recognition of DNA, which will be blue in the next slide, then a beta turn, then another alpha helix. These are sequence-specific binding, and that's due to the specific contacts between the recognition in the helix and the major groove. And so that's what these sort of look like. And again, not going to be super responsible for knowing these structures exactly. Just knowing the three types, I think, is fine. Yeah. Other type is zinc finger. These are about 30 amino acids, and it has this coordination site with a zinc ion there in the middle, and it makes kind of like a finger. I don't know, it never really looks like a finger to me, but they kind of point into the major groove of the DNA sequences that they recognize. Okay. And then finally, leucine zipper. These are longer alpha helices that almost make kind of like a pliers or tweezer, something like that, that squeezes the DNA. So if we continue on with bacterial gene regulation, there are a few other examples that we want to bring up here. The LAC operon, that's going to be the one that you really need to know. Some of these others, you're not going to need to know the details of, but just knowing of their existence, I think, for this class is good enough. So we looked at the lac operon. There's also a trip operon or tryptophan operon. And it also, it's an operon, so it uses a repressor. And that repressor is slightly different here than the lac operon. The repressor doesn't bind until the small molecule effector binds the repressor. So once tryptophan is present, it binds to the repressor, which causes it to bind to the operator, and that slows the expression of these genes. And these genes here are for the synthesis of tryptophan. So a little bit different. The lac operon's controlling the metabolism of lactose. The trip operon's controlling the biosynthesis of tryptophan. So if you have lots of tryptophan around, you don't need to express these genes. Right. And so this is an example of something that's regulated, what's known as transcription attenuation. Right? And that is a case where transcription can begin, but then it's halted by a stop signal, which we call an attenuator. Right? And this is relying on the fact that in bacteria, transcription and translation are typically simultaneous. Right? The attenuator sequence is in the 5' region of the leader sequence, and that it can make the ribosome stall. So the attenuator, that's it's determining whether or not transcription is going to be stopped or if transcription will continue. So the leader region, it can form stem loop type structures. Right? And this leader is 160 nucleotides long, and it has 4 segments here that are kind of color coded. Right? So if segments 3 and 4 base pair, they form a hairpin. That's the attenuator signal, that's the stop signal. If segments 2 and 3 base pair, that's where transcription proceeds. Okay? And thus no attenuation occurs. Right? Not going to be responsible for these details on the exam. It's just kind of an interesting way of controlling it. Okay? So 3, 4 bind, that makes this, that stops the transcription. 2, 3 pair is the one that allows it to go. Right, so example 3 is the regulation of the SOS response. We mentioned that briefly, the SOS response, when we talked about DNA repair mechanisms. SOS response is the response to excessive DNA damage. So damage that's very bad and you need an emergency, in a sense, to repair it. Okay. The SOS genes are repressed normally by what's known as the LEX-A repressor. So this is just an example of all these genes being repressed by LEX-A. Okay. And they bind, again, operators at several genes. When DNA is damaged, lots of single-stranded DNA is produced, and that single-stranded DNA is bound by the protein REC-A. So the REC-A is a protein we mentioned in a previous lecture in chapter 25. Eukaryotes, it's called RAD51. But REC-A's is that it's sort of like a single-stranded binding protein, and it helps, you can kind of think of it, helps stabilize a single strand, helps it get inserted into a double strand when you're doing recombination. Right? So REC-A also interacts with the LEX-A repressor, and it causes the repressors to what you can either just think of it just causes those repressors to leave. Okay. So in that sense, REC-A is single-stranded binding protein, but it's also acting like an effector that removes LEX-A. Okay, 4th example here, synthesis of ribosomal proteins. When bacteria need more protein, when they're growing cells, they make more ribosomes, right? That makes sense. If you need to make more protein, you're going to make proteins made on ribosomes, translated, so you need more ribosomes. Right, so that synthesis of ribosomes does consume energy and resources, so this is a regulated process. You don't want a bunch of ribosomes around when you don't need them, because that'd just be a waste of energy. So the R ribosomal proteins are regulated via what's known as translational feedback. So what is translational feedback? Each operon for a ribosomal protein encodes what's known as a translational repressor. The repressor binds to the messenger RNA and blocks the translation. So here's a little bit different method, right? Instead of it binding to DNA, stopping transcription, these bind to messenger RNA and stop translation. Okay. The repressor has a greater affinity for ribosomal RNA than messenger RNA. So translation is repressed only when the synthesis of ribosomal proteins exceeds a level needed to make ribosomes. Right, so that might be a little bit confusing, but if you have a lot of ribosomal RNA around, your repressor is going to bind to them, and then your messenger RNA is going to be free to make proteins. Right? Once that level equalizes, then you get repressor binding to the messenger RNA. Right? So kind of a second part to this, ribosomal RNA synthesis is regulated, but it's regulated by amino acid availability, and that's what's known as the stringent response. Okay? And that happens when amino acid concentrations are low. Right? So your lack of amino acids around, what's that going to do? Well, you're going to have tRNAs that are uncharged, meaning that they don't have amino acids attached to them, right? So if an uncharged tRNA binds to a ribosome at the A site, what's that going to do? It's going to trigger a cascade that begins with the stringent factor, which is protein REL-A. So we talked about REC-A, REL-A is a similar protein, but it's involved in this translational control. So stringent factor, the details here, don't need to know those details. You can just think of here in the A site, we have a tRNA that doesn't have an amino acid. That binds, that's going to activate this stringent factor, and that stops ultimately RNA polymerase, okay? Because you don't want to keep making messenger RNAs if you don't have the amino acids to do the translation, right? So it just stops the transcription at RNA polymerase. Right. The last kind of class here, 5, RNAs that are participating in regulation. And there's two different ways that this happens, either in a trans mode or a cis mode. So what's trans? That's when a molecule is affected by a separate molecule. And so these sRNAs follow that pattern. Cis is when a molecule affects its own function, and those are what we call riboswitches. You can get both positive and negative regulation with this. Okay? So here our sRNA is binding to a site that then, once that sRNA is bound, it opens this up for the ribosome binding site on the messenger RNA. In a negative way, you can have an sRNA that binds the ribosome binding site and thus hides it, right? So it can't be translated. Okay. And there's a specific example here. Again, not going to be responsible for this on the exam, but the ribosome binding, what we called Shine-Dalgarno sequence that we've mentioned before, that sequence is here. That is in a stem loop conformation. And then if you get this DSRA protein, or excuse me, RNA that's binding to it, it helps bind to that, and that opens up the Shine-Delgarno sequence to be exposed, so translation can occur. Okay. So riboswitches that regulate messenger RNA function in a cis action. So we saw the trans examples before. Now when we're talking about riboswitches, these are all in cis action. Okay. Binding of a ligand affects the conformation of the messenger RNA and its activity, and the riboswitch will allow the messenger RNA to participate in their own regulation. So here's an example of that, of a riboswitch here. So if you have something here which we call an aptamer, it can bind TPP in this case. This is our effector molecule here. And that will stabilize transcription terminator structure, and it aborts the transcription. So other things that this can also control are the splicing sites. So regulation of intron splicing, right? And that's kind of examples in fungal and in plant introns, but nevertheless, a kind of an example of how that can be controlled by a riboswitch. Important thing for you to know for riboswitches that it's contained within that messenger RNA, and the small molecules can bind and either stabilize the translation of that messenger RNA, or it can kind of block the translation of that messenger RNA. But they are within the same molecule. These are sort of a new area of research, developing area. They've been found to respond to many coenzymes and metabolites. So it's a way of kind of regulating gene expression based on how much metabolite concentrations you have within a cell. And they are found to regulate splicing. An interesting thing is that in bacteria, some of these riboswitches are unique to bacteria, and they present a promising target for antibiotics. Okay. The last thing we'll talk about with this examples of gene regulation is by gene recombination, but really for this, you're not going to be responsible for this on the exam. Okay? This is just a recombination process that you can remove a promoter relative to the coding sequence, or you can put genes into multiple orientations to alter the expression. So you're just like moving pieces around to change their expression. But really, I think this is pretty much unique to prokaryotes. We're not going to focus on it in this class. Right. The second part of this lecture, in probably a little bit shorter part of the lecture, I'm going to deal with eukaryotic gene regulation, and primarily transcriptional control. Okay. So the basics of eukaryotic gene regulation, we'll talk about how our eukaryotic DNA has to be sort of modified to be able to actually transcribe it, and in that modification there is a way of controlling what gets transcribed at any given time. Okay. Talk a little bit about the components of that transcriptional control, and it's all activation in eukaryotes. the effector molecules that are in play here in eukaryotic cells are hormones typically. Okay. We'll talk a little bit about translational regulation, and then at the very end just briefly mention gene regulation and development. So the features of eukaryotic gene regulation. Well, remember our picture of a chromosome in nucleosome structures, right? You have histone proteins, DNA is wrapped around it, and then it's kind of a coiled coil with these. So this structure is very compact. How do you get in there if a gene, is buried in that DNA around one of these histones? How do you get the machinery in there to actually start transcribing it? So that's what makes eukaryotic gene expression very, very complicated. Okay. That these promoters in your DNA sequence are hindered, at least the RNA polymerase there is hindered to get to them because of that structure. So to express a gene here, it requires remodeling of chromatin. Okay. And pretty much shouldn't say that, but what we'll really focus on in this class for eukaryotic regulation is positive regulation. And it turns out that this positive regulation is required to get even a basal level of gene expression, because obviously if it's your chromosome structure's left as is, you're really not going to get any gene expression in there, right? So you need these ways to activate it. So when we talk about chromatin, there's a couple terms that we use. Euchromatin is the less condensed form of chromatin. That's the one that gene can be expressed in. Heterochromatin is the one that's more ordered, more compact, and transcriptionally inactive. So how do you get genes to be active transcriptionally? Well, the nucleosome has to be repositioned. How do you do that? It's with histone variants. So those histone proteins get modified. And then you have these covalently modifications to those histone proteins. These terms not going to be super important. What you need to know from these is that they're remodeling the histones, remodeling the histones, thus restructuring, changing the orientation of the nucleosomes, and they lead to transcriptional activation. And this is done through covalent modifications. So things like methylation, phosphorylation, acetylation, those are things we talked about in the previous chapter when we talked about post-translational modifications. Those same post-translational modifications, when you do them to a histone protein, that remodels the nucleosome and allows transcription. All right, so those are the ones that we really focused on. Right. Remember phosphorylation, that occurs on amino acids with OH, alcohol side chains, so serine and threonine. Methylation, if you remember, that is occurring at lysines. So we have lysines specifically on histone 3 here. Also can occur on arginine. Okay. And the methylation recruits things known as HATs, histone acetyltransferases. That protein, or those proteins, the HATs, then acetylate lysines. And after you acetylate those lysines, that makes it have a decreased affinity for the histone, for the DNA. So the lysine on the histone, when it's acetylated, now it doesn't bind the DNA as tightly, and thus you can get I don't know if unwrapping is really the right term, but a looser interaction there so your machinery for transcription can go in. The opposite of a hat is a histone deacetylase, or HDAC. Those remove the acetylation there and thus make the chromatin inactive. So hats activate it for transcription, HDACs inactivate it. Some somewhat recent article dealing with this, lysine on histone 4, not acetylated as well in older mice, so that shows its importance, specifically here with genes that allow for memory storage. And this brings up the idea that there's a nucleosome code or histone code that must be encoded somewhere there in the genome that gives the direction for this organization, the nucleosome organization. And the nucleosomes seem to occur at very specific sequences, and then the covalent modifications seem to occur at those specific regions as well. Right, here's just another picture of what we talk about. Heterochromatins, more ordered, more compact. You don't get transcription there. Euchromatin, that's when you have these histone modifications. Again, those are things like phosphate groups, methyl groups, and acetyl groups, specifically acetyl groups on lysines. And you can get transcription, or you do get transcription in euchromatin. So RNA polymerase II, that's the RNA polymerase in eukaryotic cells that does transcription. It requires 5 types of proteins. One are transcription activators or enhancers. Those are proteins binding to upstream activator sequences. So similar to an up element in bacteria cells. Architectural regulators, those again, we kind of mentioned those near the start of the lecture, but they help bend DNA and get sequences that are further apart from each other and primary sequence closer in 3-dimensional space. You need chromatin modification, those remodeling proteins, HATs, HDACs. You need coactivators. Those are acting indirectly with other proteins, not with the DNA itself. And then you need transcription factors. So remember, we talked a little bit about this more towards prokaryotes, but the TFs are transcription factors. And so this is just a picture of this, how this is occurring, right? Not going to be responsible for these details, but you can get bending of the DNA so that your upstream elements come closer to where your promoter regions are. Okay? And if you have repressors, right, those are going to be binding to repressor sites. The control of this is going to be what's referred to as combinatorial control. So there's a combination of things at play here. Activators or enhancers are mixing and matching. Okay? So depending on the specific mix of activators, that's going to tell you which specific genes are going to be activated. Different combinations regulate different genes. So on average, 6 regulatory sites are needed for multicellular organisms for a particular gene, but up to 12 sites can be common. Co-activators, such as the mediator we saw in the previous picture, and TATA binding proteins are going to be assisting with RNA polymerase. Polymerase II here in eukaryotic cells has really no affinity for those promoter regions and that's where the mediator and the TATA binding protein come into play there. They're what help bring that polymerase II into proximity to that promoter region. Binding of these activators trigger promoters to bind RNA polymerase II. So binding of one activator seems to enable binding of additional activators. So that's kind of back to that idea of combinatorial control. Components bind in a regular order. So first one activator binds, then a second one binds and increases the affinity at another site and so forth. Histones are displaced as the activators bind. Okay? And that process here, right, because you're displacing histones here, this is coordinated with chromatin remodeling. So if we look at this, our DNA here, we're curved already. You can kind of picture, some other, protein here that's architectural protein that's curving this. Right? We have an activator site where our enhancer site, I should say, where our activator binds. That binding then can bring in our co-activators, mediator proteins that aren't binding to the DNA necessarily directly, but they're interacting with our activator. Okay? And thus they can get close to our TATA box here, this region, close to our promoter. Then our transcription factors can come into play there, brings transcription factors to that promoter region, and thus eventually the polymerase is made. Not going to need to know any of these details, so don't think you have to try to memorize all of these transcription factors here. Just the general overview of the process. So how do multicellular organisms like us get signals to tell what genes to express? Like in bacteria, you can kind of think of those coming from small molecules that are in the environment. Something like allolactose there is around when lactose is in the growth media for a bacteria. And so that tells it, okay, I can potentially start metabolizing this lactose. I can start making these genes. In a multicellular organism, it's a little bit different, right? Because the cells in the middle of our bodies, in some organ, don't necessarily interact with anything in our environment, right? So this is under control by, typically by hormones. Those are small signaling molecules that circulate through our body and go from one place to another to tell it to start expressing certain genes. Okay. There are two types we'll talk about. Type 1, hormone receptors. So the hormone's a small molecule signal. The receptor is what binds it and then does the action. So type 1 hormone receptor, they're monomeric, and these are receptors for sex hormones. They're found in the cytoplasm, and they're in complex with heat shock protein 70. When the hormone binds, heat shock 70 dissociates, so here a hormone. And hormones in general are hydrophobic enough that they can pass through plasma membranes. Okay. So it passes through that, binds to that receptor, it leaves heat shock protein out, and then dimerizes. When it dimerizes, it can be brought through to the nucleus. The nucleus there, it binds what's known as a HRE, hormone response element, binds there, coactivator binds, you can get polymerase, which then turns on the gene. Important part here, type 1 hormone receptor, monomeric, they bind lose heat shock protein, dimerize, go in, bind to HRE, turn on expression. Type 2, which an example of that is the thyroid hormone receptor. These are found in the nucleus themselves, and they're found bound to the DNA. So the hormone comes in, and you have a co-repressor bound there. So once that hormone binds, that repressor dissociates, and when that dissociates, then you can get activation of transcription. So slightly different mechanism there. In type 2, the hormone goes through the plasma membrane, through the cytosol into the nucleus itself. Type 1, it reaches a cytoplasm where it binds its receptor. That's the main difference there. Okay. They both use those hormone response elements, or HRE. Those are regions of DNA where the hormone system is working. Okay. They typically have DNA binding. The hormone receptors typically have DNA binding domains that are zinc fingers. Okay. That kind of just brings back to those three types of proteins that bind DNA. This was one of them. Okay, and those are hormone receptors. The last few slides here, I'm going to be talking about gene regulation in eukaryotic cells, and that's undergoing translational control. Not going to really go into too much details, but if you go on to study medicine or anything in developmental biology, it's really turns out that it's very important where you turn on a gene in both space and time. So not just the type of cell, but at what stage in the lifespan do you signal that gene to turn on. So we talked about these. It's very hard to see this picture, but at the beginning of this lecture, we talked about all the different places where you can control gene expression in cells. The messenger RNA stability, that's kind of what we're going to be talking a little bit more about here. Right. So translational regulation mechanisms. Translation initiation factors, those are subject to phosphorylation by kinases. That would deactivate them. Proteins binding directly to messenger RNA, that would be repression. Proteins binding to this EIF4 and RNA-mediated regulation. And the examples of those are the microRNAs or small interfering siRNAs. So what we'll really focus on in this lecture is really this fourth one is what I want to focus the most on. Translational repressors, those are proteins that bind to the messenger RNA that just kind of stop the translation to it. Okay, and that's where the EIF4 comes into play. If you stop this, you stop translation. And again, those are proteins binding directly to the messenger RNA that then make it kind of loop up, right? The messenger RNA loop up around the ribosome, and you don't get translation. All right, so moving on to that fourth point there, microRNAs. MicroRNAs are also known as gene silencers. They bind to messenger RNAs, and by binding to the messenger RNA, they prevent translation. So the gene has already been transcribed into messenger RNA, but if you block the translation of that messenger RNA, that's another level or another place of control you can have. Okay. Some of these microRNAs are made briefly during development at certain times, and they're called small temporal RNAs or sTRNAs. Basically, miRNA, siRNA, and sTRNA are all acronyms of kind of the same thing. So how does this work? Well, you'll have a precursor RNA, something like that's going to be a messenger RNA. And then you can have your small interfering RNA bind to it. Our messenger RNAs down here, I should say. These are precursors to the small temporal RNAs or siRNAs. Okay. And they're processed by a protein called dicer. So if you've ever heard of that protein dicer, that's something that's making these. Right. In the duplex form, they're kind of inactive after dicer works on them. Then they become their active form, which then can go bind. They're single-stranded now. They can bind a messenger RNA, and they either target it for degradation, or they just simply block translation from happening. Right. So that's, those in a nutshell. This is a very big area of study currently, because researchers can use these small RNAs to block gene expression. And you can block, really, if you design these, you can block any gene you want, and then you can study what the effect of that is. So that's a really good way to know, what genes do. Right, so here's an example of a silence gene from a paper. So if you have a gene of unknown function, you can silence it in cell, like a cell culture, and then the effects can be monitored, right? You can do things like qPCR, microarray, Western blot, and get an idea of what that gene is doing. And just as an aside here, I mentioned previously in the lecture, housekeeping genes. Typically, when you do a Western, here the RSK2 protein is the one that they tried to silence, and that's shown here. They turn that gene off. NC is sort of a normal control. So you see what the level that's normally at, they can turn it off. They need to ensure that you're loading approximately the same amount on each lane. So that's where this GAPDH comes in, right? That's a housekeeping gene. And you can see in all of these, it's about the same. So this RNA interference, that holds a lot of promise for silencing overexpressed genes. So in some disorders, you have a case where genes are overexpressed, and it causes a disease state. So if you can use these to silence those overexpressed genes, they can be used as therapeutics. And one in particular place where that could be is in cancer, because in cancer cells, a lot of times you get how they turn into cancer. Some of the mutations lead to overexpression of certain genes. Gene regulation, this is very important during development, and you could really spend a whole class. You can have a whole quarter-long class on just this lecture, but you could probably also have a whole class just on developmental biology, right? And how what genes are important when, and how things get differentiated and segmented. But just briefly mentioning it here, eggs are preloaded with RNAs from the mother. Okay, so they have lots of messenger RNAs already within them. An example here, you have 18,500 messenger RNAs in an egg, but only 6,000 in a mature sea urchin. So the sea urchin egg has 3 times as many messenger RNAs in it. So how that egg turns into an organism, it's based on things being segmented and differentiated at particular times. Like it all starts out as one cell, and then that cell divides. They're all similar cells, but at some point in time, one of those cells decides, okay, I need to be turned into something that's going to look like a leg, let's say, or a skeletal system or circulatory system. So all of this stuff, you're coming from the same cells, right? But you're turning into different, very specialized cells. How does that happen? Well, it happens via different gene expression. So gene expression needs to be regulated in both space and time. Okay. And one of the leading researchers in this, shown here, his work on fruit flies is sort of mentioned in the book. Not going to know you don't need to know any of those specifics for our class, but that's a very common model system for developmental biology. Why? Because they're very easy to work with. Okay. They're somewhat simple organisms, and you can, reproduce them to a large extent. So they're very easy to keep, right? Okay. Again, genes involved in this segmentation process are called gap genes or pair rule genes, and they segment polarity, segment polarity genes. Those are transcribed after fertilization. So if you ever heard gap genes, pair rule genes, and segment polarity genes, those are genes involved in segmentation. These genes are regulated by the maternally supplied messenger RNA. Okay, so that's another kind of interesting thing there. The regulation of those comes from the pre-supplied messenger RNA. So home or HOX in humans, those are genes controlling body plan in the embryo. The protein product of these is a transcription factor. It contains a DNA sequence known as a homeobox. That might have been a common term. You hear homeobox. All right, so if you ever hear that term, that is something that's involved in sort of body plan differentiation. All right. Not going to need to know these in specifics for our class, just kind of if you're interested. Okay. The last thing I'll mention here is about stem cells. Okay. So once you have fertilization of the egg, you have kind of like a bundle of cells. And those cells, right, are all similar. So again, how do they differentiate? How do they tell, okay, I'm going to turn into something that goes on to make the circulatory system or the nervous system? Well, that's comes into a point where the cells are what we call pluripotent. Okay? And these pluripotent cells then can be differentiated into different systems. So these pluripotent cells are kind of what we know as stem cells, right? And they hold a lot of promise for treating different diseases, right? Because if you can, take out some stem cells from one part of your body and put it into an organ or tissue that has suffered an injury or has some sort of a degradative, been damaged to where it's degraded, like cartilage, let's say, you can put in stem cells there. They're pluripotent, so once they get the signal from the environment on what type of cell to differentiate into, they can turn back into, let's say, cartilage and help a damaged joint. Right? So this is a burgeoning field of research, and we don't really go too much into it, but one of the things that stem cells can do is that they can renew themselves, and they can then be differentiated. So they can they're sort of self-generating, but then once you get an appropriate signal to them, they differentiate, and then they can turn into other organs or types. Okay? So ES, sometimes you'll see that abbreviation. That just stands for embryonic stem cells. So ES cells are embryonic stem cells. And again, pluripotent means that they can differentiate into any tissue. So those are the two most important things to take away from this slide. Okay. So that concludes the lecture and the quarter. Just wanted to thank you all for being a great class, and good luck on the final, and hopefully you all get good grades on that and go on to successful post-baccalaureate careers.